In this figure, we're going to assess and examine the factors that generate the difference between the intracellular fluid and the extracellular fluid, as well as what sets up the resting membrane potential, that voltage across the cell membrane. Now, this is the last panel of this particular figure, so we'll summarize everything in this one picture. And we start with the presence of cytoplasmic proteins that tend to have a net negative charge. Now, this does affect, these proteins are trapped, this does affect the distribution of other permeable ions to a slight degree. It's called the Donin equilibrium. But it is a slight degree, and so that doesn't contribute dramatically to a difference in the membrane voltage. Now, the next thing to look at, which is a huge factor in determining differences between inside and outside the cell, would be the sodium-potassium pump shown here. That sodium-potassium pump moves 3 sodium ions out for every 2 potassiums moved in for each cycle of the pump, which means that it's electrogenic. Again, that does contribute to the membrane potential, but it is very minor, so most people just ignore it, even though it's termed electrogenic. Now, what it does do very significantly is generate this very high concentration of sodium outside the cell and very high concentration of potassium inside the cell, essentially setting up very significant chemical gradients, one of the factors that drive those ions to move into or out of the cell. Now, the next thing that happens in combination with that sodium-potassium pump is the major determining factor of the membrane voltage, and that is this guy. This guy is a potassium leak channel. And what it does is it allows potassium to passively move down its chemical gradient and out of the cell. What that does is it adds positive ions immediately to the outside of the cell, but in addition, it strands anions on the inside of the cell. In other words, as a positive ion leaves this environment, it essentially leaves behind a higher concentration of negative charge. And that sets up the membrane voltage known as resting membrane potential, that difference between inside and outside in terms of charge. It's important to note this is a spatial distribution of charge across the membrane. The cytoplasm as a whole and the interstitial fluid as a whole is electroneutral. There is not a difference in the number of positive and negative charges. It's a spatial separation right here where we're sort of stranding negative charges on the inside as we add positive charges to the outside that generates the majority of the membrane potential. Okay, the only thing left now really is to look at what generates the rest of the difference between intracellular and extracellular fluid. Well, we have a chloride leak channel, right? And because the cell, because of the potassium leak, is negative on the inside, that chloride is repelled from the inside of the cell fairly significantly. So even though there's a chemical gradient inward, it's repelled strongly, and chloride tends to be pretty close to its equilibrium, balancing the chemical and electrical gradients when a cell is at its resting potential. If we come down to the bottom here, we see we have a calcium pump or a calcium ATPase that causes or generates a high concentration of calcium outside the cell. There's not really a significant leak for calcium to enter the cell, so that gradient is maintained. And then the last thing we'll point to over here is most cells have a weak sodium leak, often through a leak channel. And that does tend to diminish the sodium gradient slightly, although our pump is counteracting that. The end result of this, though, is that the membrane potential, the resting membrane potential, is largely due to potassium, but it's a little bit up north of the potassium equilibrium potential, where potassium is perfectly equal, because of this weak sodium leak. In other words, the sodium comes in and makes the cell slightly more positive than the potassium leaking would if the sodium didn't leak at all. And there you have it, the sum of differences between inside and outside the cell, as well as generation of that resting membrane potential.